I am Suganya. I am working as an assistant professor in Department of Biochemistry, KSRCAS, Tiruchengode. Today we are going to discuss about enzyme. What is enzyme? Types of enzyme. We can see. First of all, enzyme. Enzyme is a nothing but it's a biocatalyzed reactions. First of all, it is discovered by Emil Fischer in the year 1926. The enzyme was first isolated in urease. Enzymes all the enzymes are protein, but all the proteins are not enzyme. Enzymes is a biocatalyzed reactions. A catalyze as a substrate increase the velocity rate of chemical reaction without itself undergoing any change in overall the process. The enzyme enhance the rate of reaction through acid base catalysis. This is the enzyme of the definition. Next, we are going to discuss about active site. Active site is also known as active center. The active site is not a rigid in structure and shape. The active site are regarded as cleft or occupying a small region in a big enzyme molecule. Enzyme that substrate bind in the active site. This is called active site. Next, we are going to discuss about 6 major classification of enzymes are involved. First of all, oxidoreductase, transferase, hydrolase, lyase, isomerase, ligase. First we are going to discuss about oxidoreductase. Oxidoreductase enzyme that catalyze the transfer of a functional group. For example, alcohol dehydrogenase, cytochrome oxidase. Here AH2 plus B, it gives A plus BH2. This is oxidoreductase. Next, transferase enzyme that catalyzes the transfer of a functional group. A X plus B, it gives A plus B and X. For the example, aldose, enamorase, enolase. Next, hydrolase. Hydrolase that brings hydrolysis of various compounds. For example, lipase, pepsin, urease. For the example, A, B plus H2O, it gives AH plus BOH. This is hydrolase. Next, lyase. Enzyme specialized in addition or removal of water, ammonia and CO2, etc. For the example, aldolase, enomerase, fumase, etc. A, B plus H, X, Y, it gives AX and BY. This is hydrolase. Next, isomerase. Isomerase involved in the isomerization of the reactions. For the example, triose phosphate isomerase. Here the A, it gives A dash. And then last, ligase. Enzyme catalyzes the synthetic reaction where two molecules are joined together in ATP is used. For the example, thiokinase. Next, we are going to discuss about isoenzyme. Isoenzyme is nothing but which differ physical and chemical properties and catalyze the same reaction of this enzyme. Isoenzyme are produced by a single gene and some may result more than one gene. Isoenzymes can be separated by 3 different ways. First, heat inactivation, chemical inhibition, and then electrophoretic techniques. This is the enzyme separation, isoenzyme separation. Next, mechanism of enzyme actions. The enzyme, it's combines with the substrate, gives enzyme substrate complex. Again, it gives enzyme plus product. We got a final product, enzyme plus product. Here, the enzyme catalysis is that substrate S that combine with E enzyme. The active site 2 from the enzyme substrate complex, that is ES. Finally, we get E plus P, enzyme and then product. A few theories have been put forth to explain this mechanism, enzyme substrate complex formation. This is the mechanism of enzyme actions. Next. Here, lock and key model. Here, the mechanism, 2 types of. First one, lock and key and induced fit hypothesis. First, we are going to discuss about lock and key model. Here, this theory first proposed by Emil Fischer. According to this model, induced lock and key model, structure and conformation of this enzyme rigid. The substrate fits to the binding site just as a key fits into the proper lock and to the hand into the proper glove. This model totally fails to explain many facts enzymatic reaction. The most behind the effect of allosteric modulators. First, the key, it combines with enzyme, it gives enzyme substrate complex, it gives enzymes. Finally, we get enzyme. This is the lock and key mechanism. Next, we are going to discuss about induced fit hypothesis. Here, the proposed course land in the year 1958. Here, enzymes, this model, the active site is not a rigid and a pre-shaped structure in induced fit hypothesis. In the interaction of the substrate with the enzyme induced fit or a conformational change that this enzyme resulting in the conformation of strong substrate binding site. Amino acids of the enzymes active site and bring out the catalysis. For like enzyme substrate complex, it's bind to the active site. This is induced fit hypothesis. Next, enzyme specificity. Enzymes are a highly specific their actions when compared with the chemical catalysis. There are four types of specific enzyme specificity. First one, absolute specificity. Second, group specificity. Third, reaction specificity, and then final substrate specificity. Here, the enzyme complementary with active site, and after that denaturation it gives denaturation incomplete active site formation is formed in the enzyme specificity. Effect of enzyme activity. Here, four main types of the enzyme activity is at pH, temperature, enzyme concentration and substrate concentration. First, we are going to discuss about this enzyme activity. Here, increase the hydrogen ion concentration is increased. The optimum temperature is 40 degrees Celsius. This is called optimum temperature. Below and above this pH, the enzyme activity is much lower, and at extreme pH, the enzyme become totally induced. Next, effect of temperature on enzyme activity. Here, velocity of an enzyme reaction increase, automatically the temperature also increase in the maximum declines. Next, the temperature coefficient is Q10, and then majority of the enzyme Q10 is to between 0 degrees Celsius to and 40 degrees Celsius, and then increase in temperature results in higher activation of the molecules and more molecules, I mean molecules and substrate. Interaction of the reaction. Here, the effect of temperature in the enzyme activity. Next, effect of substrate concentration on the enzyme activity. Here, increase the substrate concentration increase, velocity enzyme also increase. Effect of enzyme concentration on the enzyme activity. As the concentration of enzyme is increased, the velocity of the reaction also increased. In fact, this property of enzyme is made used in determining the serum enzymes for the diagnosis of disease by using a serum keeping all the other factors like substrate, pH and temperature. This is also factors affecting enzyme activity. Students, today we discuss about enzyme introduction, then types of enzyme, then factors affecting enzyme activity and also active site. So, next class we are going to discuss about what is allosteric enzyme. Thank you.